Welcome guys to my YouTube channel Study with Dr. Brainiac. Today we will discuss chapter vitamin from the textbook Pankaj Naik 4th edition. In this chapter we will see introduction of vitamins, definition and classification of vitamins, water soluble vitamins, fat soluble vitamins and their summary. Let's begin with our topic. So introduction. The name vitamin was proposed in 1911 by Polish chemist Casimir Funk for the nutrient compound required to prevent the nutritional deficiency disease because of its vital need and because chemically it was found to be an amine. Later, after a number of other essential organic nutrients were discovered, the E was dropped. When it was found that not all of them are amines, the term vitamin has now been adopted universally and applied to a group of biologically essential compounds to include 14 compounds which cannot be synthesized by human beings. They must therefore be supplied through food. Since their chemical nature was unknown, later designations were applied for their nomenclature. Example: vitamin A, B, C. Later, vitamin B was shown to consist of several substances and subscripts were added, that is vitamin B1, B2, B6, etc. and collectively called vitamin B complex. Definition and classification of vitamins. Vitamins are organic compounds that are required in small quantities in organic and milligram quantity per day for a variety of biochemical functions and for which generally cannot be synthesized by the body and must therefore be supplied by the diet. Some can be synthesized by intestinal microorganisms, but in quantities that are not sufficient to meet our need. They may be water or fat soluble. Classification of vitamins. They are grouped into two categories based on their solubility: water soluble vitamins, fat soluble vitamins. Now take the water soluble vitamins. It contains two groups: vitamin B complex, vitamin C, or ascorbic acid. Vitamin B complex has so many vitamins in them. These are vitamin B1, B2, B3, B5, B6, biotin, folic acid, vitamin B12. Fat soluble vitamins, which include vitamin A or retinol, vitamin D, cholecalciferol, vitamin E or tocopherol, vitamin K. Here is the table which summarizes the various vitamins. And first we will see water soluble vitamins. Thiamine, which is vitamin B1, active form TPP. Source: cereals, meat, nuts, green vegetable, egg. Daily requirement 1.1 to 1.5 milligram. Function: coenzyme for oxidative decarboxylation and transketolase reaction. Deficiency: very Wernicke-Korsakoff syndrome. Wernicke-Korsakoff syndrome mostly seen in chronic alcoholics. Riboflavin, vitamin B2, FMN, FADR, active form. Source: yeast, germinating seed, green leafy vegetable, milk, egg, liver, meat. Daily requirement 1.3 to 1.7 milligram. Function: coenzyme for oxidation reduction reaction. Deficiency manifestation: chiliosis, glossitis, dermatitis, vascularization of cornea. Niacin, vitamin B3, NAD positive, NADP positive. Source: legume, yeast, liver, meat. Daily requirement 15 to 20 milligram. Functions: coenzyme for oxidation reduction reaction. Deficiency manifestation: pellagra. Pantothanic acid, vitamin B5, active form coenzyme A, CoASH. Source: wheat germ, cereals, yeast, liver, eggs. Daily requirement 5 to 10 milligram. Functions: acyl carrier. Deficiency manifestation: burning feet syndrome. Pyridoxin, vitamin B6, PLP, active form. Source: yeast, unrefined cereals, pulses, vegetable, meat, fish, egg yolk. Daily requirement 1.6 to 2 milligram. Function: coenzyme for transamination decarboxylation, non-oxidative deamination, transsulfuration reaction. Deficiency manifestation: epileptic convulsion, dermatitis, hypochromic microcytic anemia. Biotin, vitamin B7, active form biocytin, enzyme bound biotin. Source: liver, kidney, egg yolk, vegetable. Daily requirement 150 to 300 microgram. Functions: coenzyme for carboxylation reaction. Deficiency manifestation: rare dermatitis. Folic acid, vitamin B9, tetrahydrofolic acid, is active form. Source: green leafy vegetable, liver, yeast. Daily requirement 200 microgram. Function: carrier of 1 carbon unit, synthesis of methionine, purine and pyrimidine. Deficiency manifestation: megaloblastic anemia, neural tube defect. Cyanocobalamin, vitamin B12. Methylcobalamin, deoxyadenosylcobalamin. These two are active forms: methylcobalamin, deoxyadenosylcobalamin. Source: only animal origin, meat, egg, liver, fish. Daily requirement 3 microgram. Function: coenzyme for reactions homocysteine to methionine, methylmalonyl coenzyme A to succinyl coenzyme A. Deficiency manifestation: pernicious anemia. Megaloblastic anemia, neuropathy, methylmalonic aciduria. Ascorbic acid, citrus fruit, amla leaf vegetable are the sources and active form. Ascorbic acid, daily requirement 60 to 70 mg. Functions: antioxidant, involved in hydroxylation reaction in the synthesis of collagen, steroid hormone, adrenaline, etc. Facilitate absorption of iron from intestine. Deficiency: scurvy. Fat soluble vitamin. Table for fat soluble vitamin. Fat soluble vitamins are vitamin A, D, E, K. So let's see the vitamin A. Retinol is another name. Active form are retinoic acid. All transforms are active form. Okay. Source: fish liver oil, milk product, green leafy vegetable, carrot, yellow and red fruit. Daily requirement 800 to 100 retinol equivalent. Functions: retinol and retinol are involved in vision. Retinoic acid regulates the expression of gene during growth and development. Antioxidant. Deficiency manifestation: night blindness, xerophthalmia, formation of white spot, dry, rough and scaly skin, retardation of growth in children. Vitamin D, cholecalciferol, only vitamin which acts as a hormone. Active form is 125 dihydroxycholecalciferol. Source: cod liver oil, sunlight induced in synthesis of vitamin D3 in skin and egg yolk. Daily requirement 200 to 400 international unit. Regulation of plasma level of calcium and phosphorus, calcification of bone. Deficiency: rickets in children or stimulation in adult. Vitamin E, alpha tocopherol. Active form is alpha tocopherol. Source: soya and corn oils, germ oil, fish oil, egg, alfalfa. Daily requirement 8 to 10 mg. Function: natural antioxidant and act as a scavenger of free radical, protect the RBC from hemolysis, prevent peroxidation of pupa in cell membrane. Deficiency manifestation: hemolytic anemia, reticular fibroplasia and premature infant. Vitamin K, phylloquinone, menaquinone. Phylloquinone, vitamin K1, menaquinone, vitamin K2. There is also vitamin K3. Source: green leafy vegetable, tomato, cheese, meat, egg yolk. Daily requirement 70 to 140 microgram. Functions: required for activation of blood clotting factor, required for gamma carboxylation of glutamic acid, residue in clotting and osteocalcin protein. Deficiency manifestation: hemorrhagic disorder, increased clotting time. Now look at the difference between fat soluble and water soluble vitamins. Fat soluble vitamin function as a precursor, coenzyme and antioxidant, while fat soluble vitamin function as a coenzyme, hormone, antioxidants. Fat soluble vitamins are usually non-toxic since excess amount of these vitamins are excreted in the urine, while fat soluble vitamins are toxic and even lethal when they are taken in excessive amount. Okay, water soluble vitamins are not stored extensively except vitamin B12, and so their intake has to be more frequent than that of the fat soluble vitamins which are stored. Only the fat soluble vitamins are stored, and in water soluble only vitamin B12 is stored. Fat soluble vitamins are stored and vitamin B12 are stored. Okay. Water soluble vitamin. Let's see the water soluble vitamins. What are the water soluble vitamins? B complex and C. In B complex, vitamin B12 is stored and other are excreted through water, through urine. Okay. Water soluble vitamin thiamine. Vitamin B1 structure. Thiamine consists of pyrimidine ring attached to a thiazole ring by methylene bridge. Look at the figure. Thiamine pyrophosphate structure, which is active form of vitamin B1, or structure of thiamine where one pyrimidine ring and pyrimidine ring, where is pyrimidine ring and thiazole ring attached with a methylene bridge. Okay, here thiamine ring attached with pyrophosphate group form the thiamine pyrophosphate. Okay, now come to the active form of thiamine pyrophosphate. Already we have seen. Source: it is present in all natural foods, but particularly good dietary sources are unrefined cereals, meat, nuts, green leafy vegetable, eggs. And white bread and polished rice are very poor sources of vitamin thiamine. Function: thiamine is required for carbohydrate metabolism. Thiamine pyrophosphate is a coenzyme involved in several enzymatic reactions, mainly oxidative decarboxylation, transketolase reaction as follows. TPP is a coenzyme for pyruvate dehydrogenase complex, which catalyzes the conversion of pyruvate into acetyl-CoA by oxidative decarboxylation. Acetyl-CoA is a precursor for the synthesis of neurotransmitter acetylcholine and also for the synthesis of myelin. Thus, thiamine is required for normal functioning of the nervous system. Thiamine pyrophosphate (TPP) is a coenzyme for alpha-ketoglutarate dehydrogenase, which catalyzes the conversion of alpha-ketoglutarate to succinyl coenzyme A in TCA cycle. TPP is a coenzyme for the enzyme transketolase in the pentose phosphate pathway of glucose oxidation. Nutritional requirement: Nutritional Research Council recommends the daily intake and 1 to 1.5 mg of thiamine for adult, which is with increased muscular activity, dietary carbohydrate, and in pregnancy and lactation. Deficiency manifestation: The deficiency of vitamin B1 results in condition called beriberi. Deficiency of thiamine occur in population who consume exclusively polished rice as a staple food. Polishing of rice removes thiamine. The early symptoms of thiamine deficiency are anorexia, nausea, mental confusion, peripheral neuritis, muscle fatigue, and irritability. Thiamine deficiency leads to three types of beriberi. Named as dry beriberi, wet beriberi, infantile beriberi. And for the latest edition, there is another name added to these beriberi. These are dry beriberi, neuratic beriberi, wet beriberi, cardiac beriberi, and infantile beriberi. Dry beriberi, neuratic beriberi. It develops when the diet chronically contains slightly less than thiamine requirement. This form of beriberi characterized primarily by peripheral neuritis, severe muscular weakness, and fatigue. And the symptoms of dry beriberi include dry skin, mental confusion, poor appetite. Wet beriberi, cardiac beriberi. It develops when the deficiency is more severe, in which cardiovascular system is affected in addition to neurological system. Means cardiovascular system plus neurological system are affected. Wet beriberi is characterized primarily by edema of extremities, heart enlargement, cardiac insufficiency. Other symptoms include tachycardia, bradycardia, palpitation. Both forms of beriberi may overlap to a varying degree, and patient of beriberi may die due to heart failure if not treated. Infantile beriberi. Infantile beriberi is observed in breastfed infant born to mother suffering from thiamine deficiency. The breast milk of this mother is deficient in thiamine. It is characterized by cardiac dilation, enlargement of heart, tachycardia, convulsion, edema, and GI disturbance, such as vomiting, abdominal colic, etc. In acute condition, the infant may die due to cardiac failure, Von-Korsakoff syndrome. It is also known as cerebral beriberi and mostly seen in alcoholics. Most important syndrome. In chronic alcoholics, the nutritional deficiency results from either poor intake of food or malabsorption of nutrients from intestine. Von-Korsakoff syndrome is characterized by anorexia, nausea, vomiting, nystagmus, depression, ataxia, loss of memory, mental confusion, peripheral paralysis, muscular weakness. Again, anorexia, nausea, vomiting, nystagmus, anorexia, nausea, vomiting, nystagmus, depression, ataxia, loss of memory, mental confusion, peripheral paralysis, muscular weakness. Antimetabolites. Thiamine can be described by thiamine is found in raw fish and seafood. Thiamine assay. Thiamine is measured by thiamine whole blood or erythrocyte transcriptase requiring TPP as a coenzyme. Activity is used to measure the thiamine deficiency. Now come to the second vitamin, vitamin B12, also known by the name of riboflavin. Structure. Riboflavin is a yellow compound. Flavus means yellow in Latin. Okay, consisting of isoalloxazine ring with ribitol sugar alcohol side chain. Riboflavin is relatively heat stable but decomposes in the presence of visible light, photosensitive. Heat stable but photosensitive. Active form of riboflavin. Active form of riboflavin. The active or coenzyme form of riboflavin are flavin mononucleotide and flavin adenine dinucleotide. Source. The main dietary source of riboflavin are yeast, germinating seed, green leafy vegetable, milk and milk product, egg, liver, meat. Cereals are poor source. Functions. Riboflavin is a precursor of coenzyme FMN and FAD, which are required by several oxidation reduction in metabolism. FMN and FAD are serve as a coenzyme for oxidative reductase enzyme involved in carbohydrate, protein, lipid, nucleic acid metabolism and electron transport chain. Some examples are given in table 7.2. Flavopod. Look at the table 7.2. Example of enzyme requiring FMN or FAD as a coenzyme and reactions where they are involved. Flavoprotein enzyme. Pathway and reaction. Amino acid oxidase, deaminase of amino acid, xanthine oxidase, purine degradation, succinate dehydrogenase citric acid cycle, acyl coenzyme dehydrogenase fatty acid oxidation, NADH dehydrogenase respiratory chain into mitochondria, pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase oxidation, decarboxylation of pyruvate and alpha ketoglutarate. Source. The main dietary source of riboflavin are yeast, germinating seed, green leafy vegetable, milk and milk product. Okay, these are the sources of riboflavin. Come to the functions. After the table, now come to this point. It is needed for maintenance of mucosal, epithelial and ocular tissue. They are also involved in protection against peroxidation in metabolism of xenobiotics. Look at the structure of riboflavin and its coenzyme FMN and FAD. Riboflavin, ribitol attached with the flavin ring. Okay, isoalloxazine ring. Flavin ring also known by the name of isoalloxazine ring. Flavin mononucleotide attached with the phosphate group. Okay, next nutritional requirement RDA for vitamin B12 is B2 for vitamin B2 is 1.32 1.7 milligram for adult. It is related to protein use and increase during growth, pregnancy, lactation, wound healing. Deficiency manifestation. Riboflavin deficiency is quite rare as it has a wide distribution in foodstuffs. It is usually seen along with the deficiency of other vitamin of B complex group. It is most commonly seen in chronic alcoholics. The characteristic symptoms of riboflavin deficiency are chiliosis, glossitis, dermatitis, vascularization of cornea, chiliosis, fissure at the angle of mouth, glossitis, inflammation of tongue that becomes swollen and magenta color, dermatitis, rough and scaly skin. Riboflavin assay, a commonly used method for assessment of riboflavin status, use the determination of FAD-dependent glutathione reductase activity in fresh lyse erythrocyte. Niacin. Come to the third vitamin B3 of water soluble vitamin, group of water soluble vitamin. Third vitamin of B complex, vitamin B3, niacin. Structure. Niacin is general name for nicotinic acid and nicotinamide, either of which may act as a source of vitamin in the diet. Niacin is a simple derivative of pyridine. Niacin is a simple derivative of pyridine. Active form of niacin are NAD positive, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, NADP positive. Figure 7.4. Look at the figure. Nicotinic acid, nicotinamide. Okay, either of them used as a vitamin. Okay, either of them used as a vitamin. Look at the figure of NAD positive. Okay, nicotinamide ribose attached with the ribose sugar. Okay, adenine attached with the ribose with the 2-phosphate group. Okay, here nicotinamide adenine dinucleotide phosphate attached with another phosphate group. Okay, look at the sources of niacin. Yeast, liver, legume, meat are major source of niacin. Limited quantity of niacin can also obtained from the metabolism of tryptophan. For every 60 mg of tryptophan, 1 mg equivalent of niacin can be generated. Okay, function. Niacin is a precursor of nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. NADP positive are involved in various oxidation reduction reactions catalyzed by dehydrogenase in metabolism. They are therefore involved in many metabolic pathways of carbohydrate, lipid, protein. Generally, NAD positive linked dehydrogenase catalyze oxidation reduction reaction in oxidative pathways, citric acid cycle and glycolysis. Whereas NADP positive linked dehydrogenase or reductase are often found in pathways concerned with reductive synthesis. Example synthesis of cholesterol, fatty acid and pentose phosphate pathway. Selected examples of enzyme and the reaction they catalyze are given in table 7.3. Look at the table. Example of enzyme requiring NAD positive or NADP positive or NADPH and reaction where they involve NAD dependent. Glyceraldehyde 3-phosphate dehydrogenase requiring glycolysis. Glyceraldehyde 3-phosphate 2,1,3-bisphosphoglycerate. Pyruvate dehydrogenase oxidative decarboxylation of pyruvate to acetyl CoA. Alpha ketoglutarate dehydrogenase TCA cycle alpha ketoglutarate to succinyl CoA. Beta hydroxy acyl CoA beta oxidation of fatty acid beta hydroxyl acyl CoA to beta keto acyl CoA. NADP dependent glucose 6-phosphate dehydrogenase for pentose phosphate pathway glucose 6-phosphate to 6-phosphogluconolactone malic enzyme transfer of acetyl CoA from mitochondria to cytosol. NADPH dependent 3-ketoacyl reductase fatty acid synthesis 3-ketoacyl enzyme to 3-hydroxyacyl enzyme HMG CoA reductase cholesterol synthesis HMG CoA to mevalonate. Okay, Come to the nutritional requirement of niacin. Nutritional requirement of niacin. The RDA 15 to 20 milligram. Tryptophan can only provide about 10% of the total niacin requirement. Deficiency manifestation pellagra. Deficiency of niacin in human causes pellagra, a disease involving the skin, GIT, CNS. The symptom of pellagra characterized by 3D. Now there are 60s are there. So dermatitis, diarrhea, dementia, death. Dermatitis, skin inflammation is seen in any area exposed to direct sunlight. Diarrhea, frequent diarrhea, nausea, vomiting, anorexia are the disorder of GI tract. Dementia, loss of memory is associated with degeneration of nervous tissue. To produce niacin deficiency, diet must be poor in both available niacin and tryptophan. Niacin deficiency occur in population dependent on maize, sorghum as a staple food diet. Deficiency of vitamin B6 pyridoxal phosphate lead to niacin deficiency as it is involved as a coenzyme in the pathway of synthesis of niacin from tryptophan. Malignant carcinoid syndrome, in which tryptophan metabolism is diverted to formation of serotonin. In Hartnupp's disease, a genetic disorder in which tryptophan absorption and transportation is impaired. Therapeutic use of niacin. Nicotinic acid used at a high dose 1 to 2 grams per day has been shown to lower total cholesterol, LDL cholesterol, VDL triglyceride in patients with hyperlipoproteinemia. Toxicity. High intake of vitamin has undesirable side effects, mainly vasodilation and flushing of liver and liver damage. Now come to the mark this point. Symptoms of pellagra. Now come to the another vitamin, pantothanic acid. Vitamin B5. The name pantothanic acid is derived from Greek word pantothen, means from everywhere, and gives an indication of wide distribution of the vitamin in food. Structure. Pantothenic is formed by a combination of pantoic acid and beta-alanine. Beta-alanine combined with pantoic acid. Active form of pantothenic acid, a coenzyme A acyl carrier protein. To form our active coenzyme A acyl carrier protein. Source: egg, liver, yeast, wheat, germ cell, etc. are important sources of pantothenic acid, although the vitamin is widely distributed. Functions. Pantothenic acid is a component of coenzyme A, coenzyme SH and acyl carrier protein. The thiol group of coenzyme A, SH and ACP act as a carrier of acyl group. Coenzyme A participates in reaction concerned with reaction of citric acid cycle. Pantothenic acid is a component of coenzyme A and acyl carrier protein. The thiol group of coenzyme SH and ACP act as a carrier of acyl group. Coenzyme A participates in reaction concerned with reaction of citric acid cycle, fatty acid synthesis and oxidation, synthesis of cholesterol, utilization of ketone body. ACP participates in reaction concerned with fatty acid synthesis, Nutritional requirement. The idea of pantothanic acid is not well established. At daily intake of about 5 to 10 mg is advised for adult. Deficiency manifestation. No clear-cut case of pantothanic acid deficiency has been reported, except in malnourished prisoners of war. In the East in 1940s, where neurological condition known as burning feet syndrome was reported and ascribed to pantothanic acid deficiency, as these people were severely malnourished and were deficient in other vitamins as well. It is not possible to attribute this specific effect to pantothanic acid deficiency. Clinical sign is observed in experimentally induced deficiency are paresthesia, headache, dizziness, gastrointestinal malfunction. Again, paresthesia, headache, dizziness, gastrointestinal malfunction. Okay, now come to the another vitamin B6, pyridoxin. Structure. Vitamin B6 consists of a maximum 3 different closely related pyridine derivatives. Pyridoxin, pyridoxal, pyridoxamine. Pyridoxin, pyridoxal, pyridoxamine. All three have equal vitamin activity as they can be interconverted in the body. These are interconverted in the body. Active form of vitamin B6 is pyridoxal phosphate. Okay, is the active form of vitamin B6. PLP is formed from phosphorylation of all three forms of vitamin B6. So source, look at the structures of pyridoxin having alcohol group. Now come to the pyridoxal and pyridoxamine. Okay. Source, pyridoxine occur mainly in plant, whereas pyridoxal and pyridoxamine are animal product present in animal product. Okay, pyridoxine in plants and pyridoxamine in animal product. Major dietary source of vitamin B6 are yeast and refined cereals, pulses, meat, poultry, fish, potato and vegetable. Dairy product and grains contribute lesser amount. Functions. Active form of vitamin B6, pyridoxal phosphate act as a coenzyme in large number of fractions of amino acid metabolism. For example, transamination, decarboxylation, non-oxidative deamination, transsulfuration, condensation, reaction of amino acid. Now come to the transamination reaction. Transamination reaction are catalyzed by transaminase and PLP act as a coenzyme converting amino acid to keto acid. Example, aspartate transaminase to alanine transaminase. Decarboxylation reaction. This is important vitamin, may be asked in university exams. Decarboxylation reaction. PLP acts as a coenzyme in decarboxylation of some amino acid. The amino acids are decarboxylated to corresponding amines. The important biogenic amine synthesized by PLP decarboxylation are given below. Gamma aminobutyric acid, GABA. It is an inhibitory neurotransmitter derived from glutamate. On decarboxylation, hence in vitamin B6 deficiency and underproduction of GABA lead to convulsions in infant and children. Serotonin and melatonin. These are produced from tryptophan. Serotonin is a neurotransmitter and stimulates cerebral activity. Melatonin is a sleep-inducing substance and is involved in regulation of circadian rhythm of body. Histamine. Histamine produced by decarboxylation of histidine. Vasodilator. It is a vasodilator and lower blood pressure involved in allergic reactions. Catecholamine, dopamine, norepinephrine, epinephrine. Synthesis of catecholamine from tyrosine require PLP-dependent dopa decarboxylase. Catecholamine are neurotransmitters and involve in metabolic and nervous regulation. Non-oxidative deamination, hydroxyl group containing amino acid serine threonine are non-oxidatively deaminated to alpha-keto acid and ammonia, which require PLP transsulfuration reaction. PLP is a coenzyme for cysteothionein synthase. Cysteothionein synthase involved in synthesis of cysteine from methionine. In this reaction, transfer of sulfur from methionine to serine occur to produce cysteine. Condensation reaction. Pyridoxal phosphate is required for condensation reaction of L-glycine and succinyl coenzyme A to form delta aminolevulinic acid, a precursor of heme. Other PLP dependent reaction. Pyridoxal phosphate is required for niacin coenzyme ND positive NADP positive synthesis from tryptophan. PLP required for synthesis of serine from glycine. The enzyme glycogen phosphorylase contain covalently bound PLP pyridoxal phosphate. PLP is required for the synthesis of sphingosine, a component of sphingomyelin. Nutritional requirement 1.6 to 2 mg. Requirement increased during pregnancy and lactation. Deficiency manifestation as pyridoxine occur in most of foods. The dietary deficiency of vitamin 6 is rare. The main clinical symptoms of deficiency are vitamin B6 deficiency causes neurological disorders such as depression, nervousness, irritability. These symptoms are due to decreased production of neurotransmitter catecholamine, GABA, and serotonin. Severe deficiency of pyridoxine causes epileptic seizure in infants due to reduced production of GABA. Demyelination of nerves causes peripheral neuropathy since vitamin B6 is required for synthesis of sphingolipid need for myelin formation. Vitamin B6 deficiency causes hypochromic microcytic anemia due to decreased heme synthesis since PLP is required for synthesis of heme. The commonest cause of pyridoxine deficiency is drug antagonists. Example: isoniazid used in treatment of tuberculosis and penicillamide used in treatment of Wilson disease. An rheumatoid arthritis can combine with pyridoxal phosphate, forming an active derivative with pyridoxal phosphate. Alcoholics may be deficient owing to metabolism of ethanol to acetaldehyde, which stimulate hydrolysis of the phosphate of the pyridoxal phosphate. Vitamin B6 activity of blood transaminases have been used frequently as indirect measurement of vitamin B6 status. Erythrocyte level of aspartate and alanine amylase transferase. Erythrocyte level of aspartate and alanine aminotransferase provide better information on vitamin B6 status. Therapeutic use. Pyridoxine is used in treatment of seizure, Down syndrome, autism, a psychiatric disorder of childhood, premenstrual tension syndrome. Toxicity. Pyridoxine seems to be safe at level of 100 to 150 mg, but taking 500 to 5000 mg per day has shown peripheral neuropathy within 1 to 3 years. Biotin. Another vitamin. Biotin was known formerly as vitamin H. We can make a mnemonic: H for hair, Deficiency causes alopecia. H for hair. Deficiency causes alopecia. It is a mnemonic for remembering the biotin. Structure: biotin is an imidazole derivative. Consists of tetrahydrothiopenene ring bound to imidazole ring and a valeric acid side chain. Source: widely distributed in food, liver, kidney, vegetable, egg yolk are the important source of biotin. Biotin is also synthesized by intestinal bacteria. Active form of biotin: enzyme-bound biotin. Biotin is an active form of biotin. Biotin is covalently bound to epsilon amino group of lysine of an enzyme to form biotin. Function: biotin is a coenzyme of carboxylase reaction, where it is a carrier of CO2. Some of the carboxylation reactions requiring biotin are given below. Conversion of acetyl-CoA into malonyl-CoA, catalyzed by acetyl-CoA carboxylase in fatty acid synthesis. Conversion of pyruvate into oxaloacetate, catalyzed by pyruvate carboxylase in gluconeogenesis. Conversion of propionyl-CoA to methylmalonyl-CoA. Conversion of propionyl-CoA to dimethylmalonyl-CoA, catalyzed by propionyl-CoA carboxylase in the pathway of conversion of propionate to succinate. It is also involved in the catabolism of branch chain amino acid, catalyzed by beta-methylcrotonyl-CoA carboxylase. Biotin independent carboxylation reaction. There are few carboxylation reactions which do not require biotin. For example, formation of carbomyl phosphate by carbomyl phosphate synthetase in urea-cycle, addition of CO2 to form C6 in purine ring, conversion of pyruvate to malate by malic enzyme. And nutrition requirement: 150 to 300 microgram recommended for adult daily intake. Biotin is synthesized by intestinal microorganisms in such a large quantity that a dietary source is probably not necessary. Synthesized by deficiency manifestations. Since biotin is widely distributed in plant and animal food, intestinal bacteria supply adequate amount of biotin. The natural deficiency of biotin is not well characterized in human. Experimentally induced symptoms of biotin deficiency are nausea, anorexia, glotitis, dermatitis, alopecia. Depression, muscular pain. Deficiency of biotin occur in the people with the unusual dietary habit of consuming large amount of uncooked eggs. Example of white contain glycoprotein. Large amount of uncooked egg. Egg white contain a glycoprotein avidin, which bind the imidazole group of biotin and prevent biotin absorption. Use of antibiotics that inhibit the growth of intestinal bacteria. Eliminate the source of biotin and lead to deficiency of biotin. Folic acid structure. Another vitamin. Folic acid, important vitamin. Structure. Folic acid consists of 3 components: pyridine ring, p-amino benzoic acid, L-glutamic acid. The component: pyridine ring, p-amino benzoic acid, L-glutamic acid. In a folic acid molecule, the number of glutamic residue vary from 1 to 7. Folic acid usually have 1 glutamic acid residue. Number of glutamic acid residue vary from usually have 1 glutamic acid residue. Structure of folic acid. You can see 3 component: tyrosine derivative power, glutamic acid, active form of folic acid, tetrahydrofolate, active form of folic acid. Folate is enzymatically reduced in two-stage process in tissue to yield a dihydro and then tetrahydrofolate, which require vitamin C. Source: folic acid is found in green leafy vegetable liver yeast. The word folate is related to folium, which means leaf in Latin. Function: tetrahydrofolate acts as a carrier for one carbon unit. The one carbon unit are methyl, methylene, methinyl formyl, formimino. One carbon unit bind to tetrahydrofolate through N5 and 10 or N both N5 and 10 position. The tetrahydrofolate coenzyme serve as an acceptor or donor of one carbon unit in a variety of reactions. Involve amino acid and nucleic acid metabolism. 5 of the major reactions in which tetrahydrofolate is given are given below. Conversion of serine to glycine. The conversion of serine to glycine is accompanied by the formation of N5 and 10 methylene tetrahydrofolate. Synthesis of thymidylate pyrimidine nucleotide. The enzyme thymidylate synthase that convert deoxyuracilate DUMP into thymidylate TMP uses N5 and 10 methylene tetrahydrofolate as the methyl donor for this reaction. Thus, folate coenzyme plays a central role in biosynthesis of nucleic acid. In catabolism of histidine, in the course of the catabolism converted into 4-amino glutamate figlu. This molecule can donate the 4-amino group to tetrahydrofolate to produce N5-4-amino tetrahydrofolate. In case of folic acid deficiency, figlu accumulate and excreted in urine. The synthesis of purine. N5-4-amyl tetrahydrofolate intermediate formed in histidine catabolic is used in the synthesis of purine and therefore in formation of protein and RNA. Synthesis of methionine from homocysteine. Homocysteine is converted to methionine in presence of N5-methyl tetrahydrofolate and vitamin B12. In this reaction, the methyl group bound to cobalamin. Vitamin B12 is transferred to homocysteine to form methionine, and the cobalamin then remove the methyl group from N5-methyl tetrahydrofolate to form tetrahydrofolate. This step is essential for liberation of tetrahydrofolate and for its repeated use in one-carbon metabolism. In vitamin B12 deficiency, conversion of N5-methyl tetrahydrofolate to free tetrahydrofolate is blocked. Nutritional requirement: the RDA of folate is 200 microgram. Requirements increases during pregnancy and lactation. RDA of folate is 200 microgram. Requirements increases during pregnancy and lactation. Okay, this figure is important on this also. Okay, folate trap. Homocysteine converted to methionine. In this figure, look at the figure 7.12. Homocysteine converted to methionine by the help of methionine synthase, and methylcobalamin parallelly converted to cobalamin during this step by methionine synthase, and cobalamin again converted to methylcobalamin by the help of N5-methyl tetrahydrofolate, which converted into tetrahydrofolate. If vitamin B12 deficiency occur at this step, so it inhibits the Formation of tetrahydrofolate from N5-methyl tetrahydrofolate, also known as folate trap. So come to the deficiency manifestations of folic acid. Folate deficiency frequently occurs particularly in pregnant women and in alcoholics. So clinical symptoms of folic acid deficiency include megaloblastic or macrocytic anemia. The deficiency of folic acid leads to impairment of synthesis of DNA, impaired DNA synthesis, impair the maturation of erythrocyte. Consequently, megaloblasts are formed instead of normoblasts. These megaloblasts are accumulated in both marrow and lead to megaloblastic anemia. Accumulation or excretion of fluid in the urine. Folate deficiency blocks the last step of retrieving catabolin due to lack of tetrahydrofolate. This results in accumulation of fluid in the body, which leads to increased excretion of fluid in urine. Look at this figure. Look at this reaction. Histidine converted to figlu, okay, or figlu converted to L-glutamate by the help of tetrahydrofolate, which in turn converted into N5-formimino tetrahydrofolate. If this step block occur in deficiency of folic acid, so figlu start excreted in urine, okay. Hyperhomocystinemia. Due to folic acid deficiency, the methylation of homocysteine to methionine is impaired, which leads to hyperhomocystinemia. Increased level of homocysteine is a risk factor for cardiovascular disease, neural tube defect in fetus. Since folate is required for the formation of neural tube in the early gestation, the folate deficiency in early stage of pregnancy increases the risk of neural tube defect. Early stage of gestation. So folate is required in the early stage of gestation to prevent the neural tube defect. Therapeutic uses: N5-methyl tetrahydrofolate called folinic acid citroverum factor is used as a therapeutic drug to overcome the folate deficiency. Cobalamin, another topic, another vitamin cobalamin. Vitamin B12, also important. Structure: vitamin B12 bear a complex cornine ring linked to cobalt atom. Cornine ring complex, cornine ring linked to cobalt atom held in the center of cornine ring by 4 coordination bond with the nitrogen of the pyrrole group. The remaining coordination bond of cobalt are linked with the nitrogen of dimethyl benzimidazole nucleotide, and sixth one is linked to either methyl or 5-deoxy adenosyl or hydroxy group to form a methyl cobalamin, adenosyl cobalamin and hydroxycobalamin respectively. Dimethyl benzimidazole nucleotide. Structure of cobalamin, either methyl or deoxy adenosyl or hydroxy group. Thus cobalamin exists in 3 forms. That differ in the nature of chemical group attached to cobalt. Cobalamin is the commercially available form of vitamin B12. Active form of vitamin B12. The active enzyme coenzyme form of vitamin B12 are methylcobalamin and deoxyadenosylcobalamin. Dietary source of vitamin B12 are of animal origin. Absent in plant food. Human obtain a small amount of vitamin B12 from the intestinal flora. Okay, absorption, transport and storage. Intestinal absorption of vitamin B12 require an intrinsic factor, a glycoprotein secreted by parietal cell of stomach. If in stomach intrinsic factor bind with the dietary vitamin B12 to form vitamin B complex, this complex bind to the specific receptor on the surface of mucosal cell of the ileum. Ileum is the site where its absorption occurs. After binding to the receptor, the bound vitamin B12 is released from the complex and enters the ileal mucosal cell through a calcium-dependent process. And its absorption is calcium-dependent process. The vitamin in mucosal cell is converted into its main plasma transport form, a methylcobalamin. It is then transported by a vitamin B12 vitamin B12 binding protein known as transcobalamin TC1 and TC2. Methylcobalamin, which is in excess, is taken up by the liver, stored in deoxyadenosyl B12 form. Storage form. Stored in liver in the form of deoxyadenosyl B12. Liver can store about 4 to 5 mg of vitamin B12 in adult, an amount sufficient to meet the body's requirement of vitamin B12 for 3 to 6 years. Vitamin B12 is the only water-soluble vitamin. That is stored in significant amount in the liver. Only the water-soluble vitamin which is stored in the liver. Function. There are only two human enzyme systems that are known to require vitamin B12 coenzyme: isomerization of methylmalonyl CoA to succinyl CoA, conversion of homocysteine to methionine. Look at the first point: isomerization of methylmalonyl CoA to succinyl CoA. Propionyl CoA is produced as catabolic end product of aliphatic amino acid and in beta oxidation of odd chain fatty acid. Produced during beta oxidation of fatty acid. Propionyl CoA produced from catabolic end of product of some aliphatic amino acid and in beta oxidation of odd chain fatty acids. The propionyl CoA is then converted to succinyl CoA. During conversion of propionyl CoA to succinyl CoA, vitamin B12 coenzyme deoxyadenosyl cobalamin is required for isomerization of L-methylmalonyl CoA to succinyl CoA. In vitamin B12 deficiency, methylmalonyl CoA accumulates and is excreted as a urine methylmalonic acid. Look at the reaction figure 7.15. Role of vitamin B12. Okay, isolation methionine, wherein these are different amino acids and odd carbon chain fatty acid come from the oxidation of odd chain fatty beta oxidation of fatty acid. Propionyl CoA form the propionyl CoA. Okay, now come to the it then further converted to L-methylmalonyl CoA, appear in urine as methylmalonate in vitamin B12 deficiency. Okay, L-methylmalonyl A in presence of methylmalonyl CoA isomer is converted to by succinyl CoA with the help of vitamin B12. If vitamin B12 is absent, it appears in urine as methylmalonate. Okay, so conversion of homocysteine to methionine. Methylcobalamin is a coenzyme in the conversion of homocysteine to methionine, which join the metabolic role of vitamin B12 and those of folic acid. This is the only mammalian reaction known to require both vitamin. Only mammalian reaction require both the vitamin. Okay, nutritional requirement. RDA for adult is 3 microgram with higher allowance for pregnancy and lactating women. Deficiency manifestation. Deficiency may arise due to decreased absorption or decreased dietary intake. Dietary deficiency is seen in strict vegetarian. Since vitamin found only in food of animal origin or in microorganisms. Deficiency of vitamin B12 lead to pernicious anemia, megaloblastic anemia, methylmalonic aciduria, neuropathy. Pernicious anemia, it is caused by deficiency of intrinsic factor in the stomach, which lead to impaired absorption of vitamin B12. It is characterized by megaloblastic anemia and low hemoglobin level with neurological disorder. Megaloblastic anemia, it occurs due to functional folate deficiency. The functional folate deficiency is seen in vitamin B12 deficiency due to folate trap. Methylmalonic aciduria, because vitamin B12 is necessary for the conversion of methylmalonic acid to succinic acid. Individuals deficient in vitamin B12 excrete excess amount of methylmalonic acid in the urine. Neuropathy, in the vitamin B12 deficiency, many neurological symptoms appear due to progressive degeneration of myelinated nerves. Degeneration of myelinated nerve is due to accumulation of L-methylmalonic CoA, which impairs the myelin sheath formation. The neurological symptoms include numbness, tingling of finger, toes, mental confusion, poor muscle coordination, and dementia. Folate trap, most important topic for first year. Short note may be asked. Methylation of homocysteine to methionine depend on vitamin B12 and 5-methyl tetrahydrofolate. When vitamin B12 is deficient and 5-methyl tetrahydrofolate cannot be converted into free tetrahydrofolate, thus most of folic acid in the body is irreversibly trapped, as it is methyl derivative of N5-methyl tetrahydrofolate. This is called folate trap. Folate trap creates a folate deficiency, and adequate supply of free tetrahydrofolate is not available for synthesis of purine and pyrimidine bases. Thus, a B12 deficiency can lead to folate deficiency. Although the tissue folate levels are adequate or high, there is a functional folate deficiency due to lack of THF, tetrahydrofolate. Again, important vitamin C, most commonly during the time of COVID, it is most used vitamin in case, because why it is used most, because it increases immunity. Okay. Vitamin C, also known as ascorbic acid, it is a 6-carbon sugar derivative. Most animals can synthesize ascorbic acid, but human cannot synthesize ascorbic acid due to lack of enzyme gluconolactone oxidase. Oxidase. Required for synthesis of ascorbic acid. Thus, humans have a dietary requirement of ascorbic acid. Look at the structure of ascorbic acid. Okay. Active form of ascorbic acid. Ascorbic acid itself is an active form. Okay. The main dietary source of vitamin C are leafy vegetable and fruit, especially citrus fruit, strawberry, tomato, spinach, potato. Cereals do not have a vitamin C. Animal tissue and dairy products are very poor source. Function: ascorbic acid functioning as a reducing agent in many metabolic processes. Function as a reducing agent. Collagen biosynthesis. Vitamin C is required for formation of collagen, where it is needed for hydroxylation of proline and lysine residue. of protocollagen. Hydroxyproline, hydroxylation are essential for the collagen cross-linking and collagen strength and stability. Since vitamin C is required for normal collagen formation, vitamin C is also involved in bone and dentition formation as well as wound healing process. Steroid synthesis in adrenal cortex. Vitamin C is involved in the hydroxylation reaction of steroid. Adrenaline synthesis in adrenal medulla. It serves as a reducing agent in hydroxylation reaction in the reaction in the synthesis of adrenaline and noradrenaline from tyrosine. Carnitine synthesis. Vitamin C function in the hydroxylation of GABA, betaine, and carnitine. Bile acid formation. Vitamin C is required for the hydroxylation of cholesterol in bile acid synthesis. Cholesterol. Look at the reaction. Cholesterol converted to 7-alpha hydroxycholesterol, then bile acid, in which vitamin C and 7-alpha hydroxylase are helpful. Degradation of tyrosine. The oxidation of para-hydroxyphenylpyruvate to homogentisate require vitamin C. The subsequent step is catalyzed by homogentisate oxidase, which is ferrous ion containing enzyme that also require vitamin C. Folate metabolism. Folic acid is converted to its active form tetrahydrofolate with the help of vitamin C. Absorption of iron. Ascorbic acid facilitates the absorption of iron from intestine by reducing it in ferrous state. Ascorbic acid is a water-soluble antioxidant. Ascorbic acid is a strong reducing agent and acts as an antioxidant. It reduces oxidized vitamin E to non-regenerated functional vitamin E. Vitamin C thought to be involved in the prevention of atherosclerosis and coronary heart disease by preventing oxidation of LDL. Antioxidant property of vitamin C is also associated with the prevention of cancer by inhibiting nitrosamine formation from naturally occurring nitrate during digestion. Nutritional requirement. The recommended daily allowance is about 60 to 70 milligram. Additional intake recommended for women during pregnancy and lactation. Deficiency manifestation. Deficiency of ascorbic acid causes scurvy. Symptoms of scurvy are related to deficient collagen formation. Function of vitamin C. Spongy swollen bleeding gums, loosening of teeth, abnormal bone development, osteoporosis, wound healing, anemia due to impaired synthesis, bruising, and bleeding due to fragile capillaries. Therapeutic use of vitamin C is preventing cold. Cancer has been scientifically supported. Although the incidence of common cold is not reduced by vitamin C, the duration of cold episode, severity of symptoms can be decreased. Toxicity. Vitamin C can be taken in dose up to 2 to 3 grams per day without undesirable effect about this liver. However, it cannot be absorbed from the intestine and can cause severe diarrhea and deposition of oxalate stone in kidney. Fat-soluble vitamins. Now the category of fat-soluble vitamins are come, which are vitamin A, vitamin D, vitamin E, vitamin K. Vitamin A, most important vitamin. Structure: vitamin A contains a single six-membered ring to which is attached an 11-carbon side chain. Vitamin A is an alcohol but can be converted to aldehyde and acid. Active form: vitamin A consists of three biologically active molecules which are collectively known as retinoids. Retinol: primary alcohol containing form, aldehyde containing form, retinol aldehyde containing form, or retinoic acid carboxyl containing form. Each of these compounds are derived from the plant precursor molecule beta-carotene, a member of family of molecules of carotenoids. Structure of vitamin A. Retinol. Look at the structure 7.7, figure 7.17. Okay. Conversion of beta-carotene provitamin to biologically active form. Beta-carotene in presence of beta-carotene dioxygenase produces two molecules of retinol aldehyde form, which by oxidation produces retinoic acid, and another reaction it produces retinol in presence of retinol aldehyde reductase. It is a reversible reaction. Retinol can be converted to aldehyde form. Okay, beta carotene, which consists of two molecules of retinol linked at their aldehyde and is also referred to as a provitamin form of vitamin A. The retinol and retinol are interconnected by enzyme retinol aldehyde reductase. The retinoic acid is formed by oxidation of retinol. The retinoic acid cannot be reduced to either retinol or retinol. Absorption, transport, and storage. Ingested beta carotene is cleaved in the intestine by beta-carotene dioxygenase to yield retinol. Retinol is reduced to retinol by retinol aldehyde reductase and NADPH requiring enzyme within the intestine. Retinol is esterified with palmitic acid incorporated into chylomicron together with dietary lipid and delivered to the liver for storage. Transport of retinol from the liver to extrahepatic tissue occur by binding of retinol to retinol binding protein. Transport of retinoic acid is accomplished by binding to albumin. Source: richest dietary source of vitamin is fish liver oil. Where are also rich source of, but meat is rather. Animal liver is also rich source, but meat is rather low in vitamin A. Other good sources are milk, dairy products, dark green leaves, spinach, yellow, red fruit, vegetables such as carrot, tomato, peaches. Okay, these sources are important. Functions of vitamin A. Vitamin A is required for a variety of functions such as vision, cell differentiation and growth, mucus secreting, maintenance of epithelial cell defect. The role of vitamin A in vision has been known throughout the study of G. Wald, who received the Nobel Prize in 1943 for his work. Wald visual cycle, important cycle. Different forms of the vitamin have different functions. Retinol and retinol are involved in vision. Retinoic acid is involved in cellular differentiation and metabolic process. Beta carotene is involved in antioxidant function. Role of vitamin A in vision. The cyclic event occur in the process of vision known as rhodopsin cycle. Wald visual cycle. Both rod and cone cells of retina contain a photoreceptor pigment in their membrane, and vitamin A is a component of these pigments. Rhodopsin, a visual purpose of the visual pigment of rod cell in the retina consists of allowances retinol bound to a protein opsin. When rhodopsin absorbs light, allowances retinol convert to all transform. The isomerization of associated with the conformational change in the protein opsin. The conformational change in opsin generates a nerve impulse that is transmitted by optic nerve to the brain. This is followed by dissociation of all retinal formed from opsin. All trans retinal is immediately isomerized by retinal isomerase to 11-cis retinol. This combines the opsin to regenerate rhodopsin and complete the visual cycle. The conversion of all trans retinal to 11-cis retinol is incomplete, and therefore remaining all trans retinol which is not converted to 11-cis retinol is converted to all trans retinol by alcohol dehydrogenase and is stored in the liver. When needed, retinol re-enters the circulation and is taken up by the retina, where it is converted to back to 11-cis retinol, which combined with opsin again from rhodopsin. Dark adaptation time. When a person enters from bright light to dark, there is a difficulty in seeing due to the depletion of rhodopsin, but after a few minutes the vision improves. During this period, rhodopsin is released and vision is improved. The time taken for regeneration of rhodopsin is known as dark adaptation time. Dark adaptation time is increased in vitamin A deficient individual. Color vision. Color vision. While vision is in dim light is mediated by rhodopsin of the rod cell, color vision is mediated by three different retinal containing pigment in the cone cell. The three pigments are called porphyrin, iodopsin, cyanopsin, and are sensitive to three essential colors: red, green, blue, respectively. All these pigments consist of 11-cis retinal bound to protein opsin. Thus, when light strikes the retina, it bleaches one or more of these pigments. Depending on the color quality of the light, the pigments are converted to all trans retinal and the protein moiety opsin is released as in the case of rhodopsin. This reaction gives rise to nerve impulse that is read out in the brain as color red if porphyrin is split green, if iodopsin is split blue, if cyanopsin is split. If the mixture of three are converted, the color read out in the brain depend on the proportion of three split. Cellular differentiation and metabolic effect. Retinoic acid is an important regulator of gene expression, especially during growth and development. Retinoic acid is essential for normal gene expression during embryonic development, such as cell differentiation and spermatogenesis, and in the differentiation of epithelial cell. Retinoic acid exerts a number of metabolic effects on tissue. These include control of biosynthesis of membrane glycoprotein and glycosaminoglycan necessary for mucus secretion. The normal mucus secretion maintains the epithelial surface moist and prevents keratinization of epithelial cell. Control of biosynthesis of cholesterol, antioxidant function. Beta-carotene is an antioxidant and may play a role in trapping peroxide free radical in tissue. The antioxidant property of lipid-soluble vitamin A may account for its possible anti-cancer activity. High level of vitamin dietary carotenoids have been associated with a decreased risk of cardiovascular disease. In nutritional requirement, the RDA of vitamin A for adult is 800 to 1000 retinol equivalent. Deficiency manifestation. Clinically, degenerative changes in eye and skin are observed with vitamin A deficiency. Effect on vision. Night blindness, nyctalopia. Night blindness is one of the earliest symptoms of vitamin A deficiency. This is characterized by the vision in night. Since dark adaptation time is increased, prolonged deficiency of vitamin A can lead to irreversible loss of visual cells. Severe vitamin A deficiency causes dryness of cornea. Severe deficiency, dryness of cornea. Conjunctiva. If this situation prolonged keratinization and ulceration of cornea takes place, this results in destruction of cornea and the cornea becomes totally opaque, resulting in permanent loss of vision. Blindness, a clinical condition termed keratinization or ophthalmia and keratomalacia are commonly observed in children. White opaque spots develop on either of cornea in vitamin A deficiency are known as white spot. Effect on skin and epithelial cell. Vitamin A deficiency causes keratinization of epithelial cell of skin, which leads to keratosis of hair follicle and rise of scaly skin. Keratinization of epithelial cell of respiratory urinary tract make them susceptible to infection. Other symptoms of vitamin A deficiency: failure of growth in children, faulty bone modeling, produce thickening cancellous spongy bone instead of thinner or more compact ones, abnormality of reproduction, including degeneration of testes, abortion, and the production of malformed offspring. Therapeutic use of vitamin A. The use of retinoic acid preparation in the treatment of psoriasis acne, several other skin diseases is related to involvement of epithelial cell differentiation and integrity. Some precancerous lesions seem to respond to treatment with carotenoids. Hypervitaminosis A. The symptoms of hypervitaminosis A include nausea, vomiting, diarrhea, loss of hair, scaly and rough skin, bone, joint pain, enlargement of liver, and loss of weight. In pregnant women, the hypervitaminosis A may cause congenital malformation and growing fetus teratogenic effect. The excess intake of carotenoid is not toxic like vitamin A. Why vitamin A is considered as a hormone? Within cell, both retinol and retinoic acid function by binding to a specific receptor protein present in nucleus of target tissue. Within cells, both retinol and retinoic acid function by binding to a specific two vitamins are considered as a hormone. One is vitamin D and vitamin A. Within cell, both the retinol and retinoic acid function by binding to a specific receptor protein present in a nucleus of target tissue. Following binding, the receptor vitamin complex interact with several genes involving growth and differentiation affect expression of these genes. In this capacity, retinol and retinoic acid are considered as a hormone. Vitamin D, again important. Vitamin D is also known as calciferol because its role in calcium metabolism and anti-rhetic factor because it prevents rickets. Vitamin D could be thought as a hormone rather than a vitamin because it can be synthesized in the body, released in the circulation. Synthesized in the body, released in the circulation, has distinct target organ. Similar to steroid action, similar to steroid hormone. It binds to a receptor of cytosol. Receptors in the cytosol. Its receptor present in the cytosol. Following binding of receptor, vitamin complex interact with the DNA to stimulate the synthesis of calcium binding protein. Structure. Vitamin D is a steroid compound. There are two forms of vitamin D. It is a steroid compound. Two forms are present. The naturally producing D3 or cholecalciferol is the form obtained from animal source in the diet or made in the skin by the action of ultraviolet light from sunlight on 7-dehydrocholesterol. It is present in our body. Artificial produced form D2 ergocalciferol is the form made in the laboratory by irradiating the plant strol ergosterol. Structure of 125 dihydrocholecalciferol, an active form of vitamin D3. Look at the structure having two hydroxyl group. Okay. Now the formation of vitamin D3 in the body: cholesterol dehydrogenation, 7-dihydrocholesterol, uvulite, and presence of uvulite form cholecalciferol vitamin D3. That is important. Figure 7.2. This is also important if you want to learn. Absorption, transport, activation of vitamin D. Exogenous or dietary vitamin D is absorbed in the duodenum along with lipid. Site of absorption: duodenum. Absorbed along with the lipid. Dietary vitamin. Okay. It is transported to the liver through chylomicron. Okay. Active form of vitamin. Cholecalciferol is an inactive form of vitamin D. It needs further metabolism to produce an active form of vitamin 125. Dihydrocholecalciferol, also known as calcitriol, is an active form of vitamin D. The steps involved in the activation are: first step, the conversion of cholecalciferol to 25-dihydrocholecalciferol. The 25 hydroxylation occur in liver catalyzed by hydroxylase. First step occur in liver catalyzed by hydroxylase. Conversion from cholecalciferol to 25-hydroxycholecalciferol. Okay. Second step, the 25-hydroxycholecalciferol form transported to kidney, where it is hydroxylated by 1-alpha-hydroxylase enzyme in the 1 position to 125-dihydroxycholecalciferol. Okay. Source: best source are cod liver oil and often fish oil. Sunlight induces synthesis of vitamin D3 in skin, egg yolk, and liver are good source. Function: vitamin D calcitriol play an essential role as a hormone in the regulation of calcium and phosphorus metabolism. It maintains the normal plasma level of calcium and phosphorus by acting on intestine, kidney, bone. Action of calcitriol on intestine: it increases the plasma calcium and phosphorus concentration by stimulating the absorption of calcium and phosphorus from intestine, by enhancing the synthesis of calcium binding protein called binding. This protein increases the calcium uptake by the intestine. Action of calcitriol on kidney: it stimulates the reabsorption of calcium and phosphorus from the kidney and decreases their excretion. Action of calcitriol on bone: it is believed that calcitriol has both anabolic and catabolic role. Calcitriol promotes the mineralization of bone by deposition of calcium and phosphorus. Calcitriol along with PTH stimulates the mobilization of calcium and phosphorus from bone by stimulating the synthesis of osteocalcin, a calcium binding protein in bone. This causes elevation of plasma calcium and phosphorus level. This figure is important. Look at the figure 7.23. In this, 7-dihydrocholesterol in presence of UV light in skin, site skin, cholecalciferol is formed from that directly cholecalciferol in liver, converted cholecalciferol converted into 25-hydroxycholecalciferol in presence of 25-hydroxylase, and then it reaches to the kidney 25 hydroxycholecalciferol in presence of 1 alpha hydroxyl is converted to 125 dihydroxycholecalciferol, also known as calcitriol, active form of vitamin D3. And then vitamin 125 dihydrocholecalciferol calcitriol reach to the intestine and bone, maintain the blood calcium, calcium and phosphorus level. Nutritional requirement. Come to the nutritional requirement. Daily requirement of vitamin D is 200 to 400. International immune deficiency manifestation. Deficiency of vitamin D causes rickets, rachitis in growing children and osteomalacia in adult. Rickets is characterized by the formation of soft and pliable bone due to poor mineralization. Calcium deficiency due to softness in the weight-bearing bone are bent and deformed. The main feature of rickets are large head with protruding forehead, pigeon chest, bow leg, curved leg, knock knee, and abnormal curvature of the spine. Rachitic children usually anemic and prone to infection. Rickets can be fatal when severe. Rickets is characterized by low plasma level of calcium and phosphorus and high alkaline phosphatase activity. Osteomalacia. The deficiency of vitamin D in adult causes osteomalacia. This condition is similar to that of rickets. Osteomalacia is characterized by the demineralization of bone. Previously formed bones. Demineralization of bone make them soft and susceptible to fracture. Renal rickets. Renal osteodystrophy. In chronic renal failure, synthesis of calcitriol in kidney is impaired. As a result, the deficiency in calcitriol occur, which lead to hypocalcemia and hyperphosphatemia. It can be treated by oral intravenous administration of calcitriol. Vitamin D resistant rickets, as the name implies, this is a disease which does not respond to treatment with vitamin D. There are various possible causes in this condition, and all involve a defect in the metabolism mechanism of 125 dihydrocholecalciferol as follows. Due to defective vitamin D receptor, due to defective 1-alpha hydroxylase activity, due to liver disease and kidney failure, as the production of 25 hydroxycholecalciferol and 125 dihydrocholecalciferol respectively will be inefficient in the damaged tissue. Hypervitaminosis D. High doses of vitamin D over a long period are toxic. And the symptoms of hypervitaminosis D include nausea, vomiting, anorexia, increased thirst, loss of weight. Hypercalcemia is seen due to increased bone resorption and intestinal absorption of calcium. The prolonged hypercalcemia causes calcification of soft tissue. Organs such as kidney may lead to formation of stones in the kidney. Therapeutic use of vitamin D analog have been used in the treatment of psoriasis. Vitamin E. Structure. Vitamin E consists of 8 naturally occurring tocopherols, of which alpha tocopherol is most active. Look at the structure of a cutter having hydroxyl group or side chain. Source, major dietary source of vitamin D: fat oils, richer source of germ oil, corn oil, fish oil, egg, lettuce, alfalfa. Absorption and transport and storage. Vitamin E is absorbed from intestine together with the dietary lipid. It is incorporated in chylomicron. It is delivered to the liver by chylomicron. The liver can be exported vitamin E into very low density lipoprotein to target cell. In the cell, tocopherols are distributed where antioxidant activity is required. The major site of vitamin E storage is adipose tissue. Site of storage, adipose tissue. Function. Vitamin E acts as a natural antioxidant by scavenging free radical molecular oxygen. Vitamin E is important for preventing peroxidation of polyunsaturated fatty acid in cell membrane. Protection of the cell membrane from oxidant is a major role of vitamin E in humans. It protects the RBCs from hemolysis. Vitamin E also helps in prevent oxidation of LDL. Oxidized LDL may be more atherogenic atherogenic than native LDL, and thus vitamin E may protect against atheromatosis, coronary heart disease. Whether vitamin E affects human fertility is unknown. In animal, vitamin E is required for normal reproduction and prevent high sterility. Nutritional requirement: a daily consumption of about 10 mg of alpha tocopherol for men, 8 mg for a woman who recommended 1 mg of alpha tocopherol is equal to 1.5 international deficiency manifestation. Vitamin E deficiency in human is rare. The major symptom of vitamin E deficiency in human is hemolytic anemia due to an increased red blood cell fragility. Another symptom of vitamin E deficiency is retrolental fibroplasia of the some premature event of low birth weight. In children with this defect, also neuropathy. Hypervitamin E. Unlike other fat-soluble vitamins such as A and D, vitamin E do not seem to have toxicity, do not have vitamin K. Another vitamin. All fat-soluble vitamins are important. Vitamin K. This vitamin is called anti-hemorrhagic factor as its deficiency produces uncontrolled hemorrhage due to the effect of blood coagulation. In 1929, Stam gave the name coagulation vitamin from the Danish word coagulation. It is also known as vitamin K structure. There are two naturally occurring forms of vitamin K. Vitamin K or phyloquinone are derived from plant. Vitamin K2 or menaquinone produced by microorganisms. Both these natural types have same general activity. Vitamin K3 guanidine is a synthetic product. Which is an alkylated form of vitamin K2. Source: excellent source of cabbage, cauliflower, spinach, and other green vegetables. Good source include tomato, cheese, dairy products, milk, egg yolk, etc. The vitamin is also synthesized by microorganisms in the intestinal tract. Absorption, transport, and storage. The naturally occurring vitamin K derivative are absorbed only in presence of bile salt. Like other lipids, it is transported to the liver in the form of chylomicron, where it is stored. Menadione synthetic vitamin K, being water soluble, is absorbed even in the absence of bile salt, passing directly into hepatic portal vein. Function of vitamin K. Vitamin K always play an important role in blood transfusion or blood coagulation. Vitamin K is required for the activation of blood clotting factor, prothrombin factor 7. One trick to remember the factors: blood clotting factor, factor 2, 7, 9, 10. These are the factors. Means, look, if we add 2 plus 7 is equals to 9. Okay, and 10. This is the trick, not the formula. Okay. Factor. These blood clotting proteins are synthesized in the liver in inactive form and are converted to active form by vitamin K dependent carboxylation reaction. In this vitamin K dependent carboxylase enzyme at the extra carboxy group at chi carbon of glutamic acid residue of inactive blood clotting factor. Vitamin K is also required for the carboxylation of glutamic acid residue osteocalcium for calcium binding protein present in bone. Anticoagulant like coumarol or warfarin are structurally similar to vitamin K, inhibit the action of vitamin K. Nutritional requirements: 70 to 140 microgram per day. Deficiency manifestation: vitamin K deficiency is associated with hemorrhagic disease. In vitamin K deficiency, clotting time of blood is increased, uncontrolled hemorrhage occur, other minor injury as a result of reduction in prothrombin, other clotting factors. Vitamin K is widely distributed in nature, and its production by the intestinal microflora ensure that dietary deficiency does not occur. Vitamin K deficiency, however, is found in patients with liver disease and biliary obstruction. Biliary obstruction limits the entry of bile salt to the intestine. Now, in newborn, because of placenta does not pass the vitamin to the fetus efficiently, and the gut is sterile immediately after birth. Following antibiotic therapy and that sterilize the gut, in fat malabsorption that impair the absorption of vitamin K, hypervitamin 6K, excessive dose of vitamin K produce hemolytic anemia and jaundice in infant. Therapeutic use and important therapeutic use of vitamin K is antidote drug that counteract the effect of poisoning to dicamol type of drug. So now here I'm signing out from the vitamins.